to receive today Professor Roberto Caminiti. Roberto is a renowned neuroanatomist, and we have been discussing and exchanging things through joint colleagues for several years, and it's really a great pleasure to meet you in real and to welcome you here. So Roberto is by training is a medical doctor with a degree from Catania. Then he started his career in Kona, and then moved to Lausanne, actually, to do his PhD under the supervision of our very esteemed colleague, Giorgio Innocenti, who passed away very unfortunately a few years ago. And this is really the connection that we have with Roberto is through Giorgio. And in 1980, Roberto moved to the US with a CNR NATO grant to the Johns Hopkins University School of Medicine. And then he came back as a faculty at the La Sapienza University in Rome, where he's a full professor and full professor of physiology at the Department of Anatomy, Histology, Forensic Medicine, and Orthopedics, very broad department indeed. And he played there several academic roles in the university, in the different schools, doctoral programs, etc. And the research of Roberto is very broad, exploring the role of different parts of the brain, many frontal and parietal cortex in alert behaving monkeys, with the goal to understand the consequences of parietal and frontal lesions on the cognitive functions mostly. And we will hear quite a lot of very interesting research that he's doing in neuroanatomy, and there is a close link also with neuroimaging, because we all have this dream of relating neuroanatomy with what we measure in imaging, and developing further the neuroimaging part, the microstructural imaging, the tractography, to have different ways to assess the brain, both ex vivo and in vivo. And that's in that context that we are having very interesting discussions that we will continue. And so, Roberto, thank you very much for being here today, and we look forward to listening to your talk. The floor is yours.
Welcome to this seminar, and thanks, Jean-Philippe. I'm very grateful for your invitation, and I'm pleased that you remember our common friend Giorgio, to whom I would like to dedicate this session and my talk, since he has been one of my main mentors, at least one person who tell me on how to look at neuroanatomy from a physiological perspective, which then resulted in a joint experiment that I did for a large part of my life. And it's also a pleasure for me to be in this lab with many people, or at least some people with whom I have collaborated, including you, Jean-Philippe, for the study on cortico-spinal connection, which is at the root of the seminar I'm going to talk here. And Giuseppe Lupino, just arrived from Parma, with whom I also did many studies on the parietal frontal system. And Giuseppe has been sort of what I consider a personal advisor, since he knows everything about the anatomy. And so the question, the sentence is, if you have a problem, ask Giuseppe. Okay, and he will answer your question. The title of my talk is Cortico-Spinal Modularity at the Roots of Action Control. This idea came to my mind based on the many collaboration that I had over time with people like you and with Giorgio, and combined to my physiological knowledge in terms of ideas and concepts derived from behavioral neurophysiology. The idea is that I want to somehow redefine and to contend is that classical neurophysiology suggests that motor cortex and its descending corticospinal tract, which you see here, play an exclusive role in action control. In other terms, motor cortex would be the only way to assess the motor periphery in an obligatory fashion before we generate the action that we can perform in a healthy life. Motor cortex for sure is important in learning of independent finger movement, which subserve and dexterity, in the distributed encoding of movement parameters such as force, direction, velocity, acceleration. And you see some example here. If you scale your force, your grip force in the wrong way, the results is that you miss your goal in adaptive motor behavior, and then in another set of function, which is selection, gating, gain control of spinal reflex. These classical views goes hand in hand with a serial processing framework in which information flow in a unique, unidirectional pipeline. First perception, then cognition, and finally action. However, this serial view is contradicted by a wealth of anatomical, physiological, and clinical data, and also fails to account for the highly parallel architecture of the cerebral cortex. And looking at M1 as the sole output of the cortex is what I consider an outdated view, which could be an obstacle for the interpretation of modern, highly complex data, such as large-scale simultaneous recording of neural activity in different cortical areas, or simultaneous recording of neural activity and both signals in animals like in the lab of Nikos Logothetis and other few people in the world. And this view can even lead to a cul-de-sac if neuroscience continues to inspire machine learning design and network modeling. On the contrast, there is evidence for the existence of different parieto-frontal spinal model that can address the motor periphery independently from M1. In other terms, without requiring to transfer information which are encoded in different, let's say, premotor or parietal or cingulate area in an obligatory fashion to motor cortex. These models observe arm reaching and its online control and tool use. Please consider that in this and the following image, these are the cortico-spinal system, which stem. This is for dorsal premotor cortex. This is for the superior parietal cortex, and the gray one that you will see in a regular fashion in all the images is the one that stems from primary motor cortex. So for each model, the independent cortico-spinal projection will be compared with the motor cortex in terms of origin and production velocities when available from the estimation of axon diameters that we did with Giorgio Innocenti, Jean-Philippe some years ago. All this said, the other model, another model concerned visomotor, conditional visomotor behavior, action plan switching, and action suppression, spatial navigation and locomotion in collaboration with the hippocampal formation, mirror mechanism, or let's say mirror system and mirror mechanisms. And this model is somehow strongly related to the lateral grasping system that Giuseppe studied for his entire life. And not only that, of course, but it also contains a mirror mechanisms for facial expression. Action initiation, motor sequence, and time encoding, which is centered on the supplementary motor area, and the defensive behavior. These are all parieto-frontal spinal model. In other terms, they have a parietal component, a frontal component, and an output system, which originate from one of these areas. Now the question concerns which are the evidence for the existence of such modularity. There are at least 5 lines of evidence that I've been able to identify to support this idea. The first one is the statistical analysis of cortical connectivity. And this is a work that I have done over the year with Alexander Battaglia-Meyer, Giuseppe Lupino, Elena Borra, and Bruno Haverbeck at NIH, and in Rome, of course. This is the monkey brain in a parcellation scheme that come from Massimo Matelli and Giuseppe Lupino, and then we also have adopted and change over time as knowledge progress. This is the medial side of the hemisphere, and this is the lateral side of the hemisphere. And the color here do not refer to individual area, but to groups of different areas. And this is the results of cluster analysis. Since cluster analysis allows defining cortical clusters, which here are differentiated by colors, and then in the brain, and that can be seen also in the consensus tree of frontal and parietal cortex here. This is a huge cluster which correspond to the dorsomedial premotor cluster, which is located here and include also this part of the brain, and include primary motor cortex, dorsal premotor cortex, supplementary motor cortex, and at a different hierarchical level, the anterior part of dorsal premotor cortex, which is also referred to area F7, and pre-supplementary motor cortex. Now, there are 2 different hierarchical levels, since these 3 areas form one cluster in which connectivity is reciprocal in most of them. In other terms, supplementary motor cortex and dorsal premotor cortex project to the motor cortex and vice versa, and they also project to the spinal cord. These 3 arrows going down means that they project to the spinal cord, while these 2 areas do not project to motor cortex, and they also do not project to the spinal cord. So the cluster analysis somehow identify groups of area which also will share different anatomical, of course, and functional properties. So clusters that you see here in different colors again, and also here in the consensus tree of frontal and parietal cortex, are composed of strongly connected cortical areas that are spatially close, in other terms adjacent, close to each other, and have strong local connectivity, since in the cerebral cortex connectivity tend to be strong locally, as is small word network of graph theory. So we consider the cluster as the basic building block of models, just building block for the moment. The third important factor of emerging from the statistical cortical connectivity is that frontal and parietal cluster are connected in a selective fashion. Here we see a projection which is mostly reciprocal from the superior parietal cortex to dorsal premotor cortex, here from inferior premotor areas to the inferior parietal lobule. And these are long oligosynaptic connections, which are mostly made by small unmyelinated fibers. The Fourth crucial question is that cluster often contain at least one area that projects to the spinal cord. There are three from this huge motor-premotor cluster. There are two emerging from area F4 and F5 C in the ventral premotor cortex. Here, there is one from the cingulate cortex, cingulate motor area, in the cingulate sulcus. There are two from the inferior parietal cortex, area PGF and IP, and there is another one from the medial bank of the superior parietal lobule, from the so-called intraparietal area, PIP. Fifth, which is also rather important, the connectivity between the parietal and the frontal cluster, shape distributed parieto-frontal system, which can be considered a central hub, which are crucial for intermodular interaction. The nature of hubs depend on the entropy that they have, at least in the formulation of the cluster analysis. Because hubs are those clusters that receive an equal number of projections from all other clusters instead from a few clusters. And for this, they are characterized by high entropy, which can be expressed in knots. And the most important cluster in the parieto-frontal system from cluster analysis, the single most important, the one which fulfill mostly these characteristics, is the cingulate cluster. The second is the motor-premotor. The third is the posterior-medial superior parietal cluster, and so on, the anterior inferior parietal. So 5 main hubs in the parieto-frontal system. What is the logic of this connectivity? The logic of this connectivity is that it is believed that this architecture minimize communication distances, then delays and energy cost. Let's now consider the ipsilateral and somehow also from time to time contralateral parieto-frontal connection, conduction velocity, and delays. One results that we have observed with Giorgio while studying this feature of axons is that the axon diameter depends on both the area of origin and termination of a given path. So you should not assume that if area X project to area Y, all the axons are of the same type, first, same diameters and conduction velocity, but they are also different from those that from area X project to area Z. So in this case, a projection which stems from the superior parietal area, which is called PEC, and which is addressed to motor cortex, dorsal premotor cortex, cingulate motor area, you can see have different axon diameters, okay, and different median conduction velocities. These cortical axons, as said, are mostly made by unmyelinated fibers, but also by thin myelinated ones, mostly those which emerge from motor-related areas, okay, and conduct action potential in a variable and temporally dispersed fashion, which requires spatial and temporal summation to discharge the postsynaptic target. In contrast, the arrival time in myelinated axons would vary by at most a few milliseconds. And these cortical axons probably play a modulatory and or a driving role. About the feature of these myelinated axons, please consider that the fact that the conduction delays are variable imply that the timing precision along axon deteriorate with increasing distances. And this loss of temporal information is particularly severe for the unmyelinated versus demyelinated axons. So that the information context, if you increase by tenfold the conduction distance, the information context, which is carried, decreased by 3.3 bit per spike. These are theoretical estimates. And for axons with long conduction distance and long conduction time, an action potential which travel at 0.3 meter per second across a 15 centimeter human brain would take over half a second to arrive at its destination, with variation in arrival time of up to 100 milliseconds. And you can imagine what is implied if you consider all these factors, especially relative to the problem of the eventual or believed necessity to transfer all motor intention, which can be formed in parietal or frontal cortex to motor cortex, instead of going directly to the spinal cord through axons like the myelinated one, which are bigger and much faster. And these are the multiplicity of corticospinal pathway that you have seen in the cluster analysis, attached to the cluster analysis in the previous slide. And this is the study that we did with Giorgio, Jean-Philippe, and Scott Nutt, I believe, in the monkey. And you see that the corticospinal system taken in an adult monkey at the cervical level is made by axons of different diameters. A few are small, medium, and also by a contingent of very, very large axons. And they convey action potential at higher speed and with precise timing code as compared to ipsilateral parietal frontal connection. Therefore, the existence of so many fast corticospinal system makes it highly improbable that all action we perform and are implemented thanks to an obligatory transfer of information from parietal information about motor intention from parietal or premotor area or singular area to motor cortex through ipsilateral slower corticocortical system. Let's have a look then at these myelinated axons, which is the evolutionary advantage of having myelinated axons. This is a nice study which considers myelinization in long-distance axons in the mammalian cortex. This is the tree shrew, the smallest mammal on Earth. You can keep it on your on your index finger. Mouse, marmoset, cat, and macaque. And then you see a transition from small myelinated, few myelinated axons to more myelinated, even more bigger myelinated axons in marmoset, cat, and macaque. And the results is that myelinization increase proportionally to brain diameters, which mean brain size and brain weight. Okay. Therefore, unmyelinated long-distance neocortical axons represent a case in which a premium is placed on speed, but they occupy significant space. While unmyelinated axons represent a case in which a premium is placed on minimization of space, but they are an accompanying penalty with a higher metabolic rate and slower speed. The separation between myelinated and unmyelinated axons in the mammalian brain occurs in this species at least at about 0.5 microns. Okay, these are the data derived from Wang and also from Lamantia and Rekich for the callosum and so on. Now, the third evidence for corticospinal modularity is the parallel activation of different cortical areas, even in distant location of the brain. And this is a study in which local field potential were recorded simultaneously from different regions of the macaque brain, from primary visual cortex, A, B, and C, along the vertical meridian of the visual field, from inferior parietal cortex, from inferior temporal cortex, from superior temporal cortex, motor cortex, dorsal premotor, frontal eye field, dorsal and ventral area 46, and polar frontal cortex, probably at the border between area 9 and 10. And the monkey was supposed to was performing a go-no-go task in a categorization paradigm. And these are the data, the raw data, but the compound data show that the earliest activation relative to stimulus evoked activity to stimulus presentation occurred in primary visual cortex between 50 and about 70 milliseconds, okay? And this is the activation time in motor cortex. It followed just in the middle of the early activation time in primary visual cortex. This is the activation time in dorsal cortex, okay, which fall within the late range of activation in primary visual cortex. And this is for the frontal eye field. This is the activation time in far prefrontal area, where the early activation overlap with the late activation in primary visual areas. And the earliest response to the visual pattern stimuli therefore appear throughout the cortex after about 50 milliseconds. And activity begin to predict the monkey choice at approximately 150 milliseconds in this paradigm, which show that passing through the entire visual hierarchy to reach the motor output is not a necessary step in visomotor system in the parietal frontal system. This is confirmed also for the parietal loci, superior and inferior parietal activation. Again, motor cortex, inferior parietal and superior parietal all fall within the range of activation, the early range of activation of primary visual cortex. For evidence for corticospinal modularity, difference of consequence of M1 lesion versus other module lesion. If M1 would be the only output of the cortex for generating action, lesion of M1 would kill almost every movement we could make. This is not the case. This is the consequence of selective lesion confined to primary motor cortex. This is what I'm talking about. Lesion confined to motor cortex, not lesion extended to subcortical structure and so on. The one that we see in the clinical picture for big stroke of the anterior cerebral artery and so on. But if you make a selective lesion in primary visual cortex relative to a control animal, what you lose is the precision grip posture and the appropriate tone, muscle tone, to retrieve a foot pellet. So the independent finger movement are impaired. Shortly after the lesion, the limb is also hypotonic. provided that the corticospinal tract exerts an excitatory influence on motor neurons, and therefore, if you remove an excitatory influence, the muscle tone decreases. Over time, this can be turned in a hypertonic syndrome, but it again depends on the extension of the lesion. For those who are interested in this kind of things, our colleague Eric Rouillet, a few miles from here in Freiburg, have been spending his life studying this kind of function, and so is like Giuseppe in Parma from a physiological perspective. So they are reference people. They are the mothers, tell the mothers for this issue. Well, this is the consequence of a superior parietal lesion. It's called optic ataxia. It's one of the component of the Balint syndrome. This patient is shown a spoon and is asked to reach at that spoon and is unable to do it. He makes variable error in a systematic fashion. But this patient is devoid of any primary sensory, visual, somatosensory, auditory, or motor deficit. Is free of these deficits, but is impaired in this specific task. Okay? And this impaired in daily life. It's not a question of being in the lab. For instance, if they want to pour some water in a glass, which is something which I would like to do now eventually, they do it like that. They put the water out of the glass. Okay? If they want to pour some sauce in the tub, they do it out. And optic ataxia also result in a delayed formation of rich trajectory, a delayed online control of movement, so that if the target shifts from the original location to another location, while under normal condition, we correct our trajectory in flight, optical ataxia patients first go to the first target and then move to the second. So they lack the nice in-flight trajectory correction. This is constructional apraxia. Constructional apraxia has been shown for the first time by Lippmann in 1908 and 1909, and this is given a copy model, which is this cross made by different blocks. These patients are unable to make a faithful copy of the model. And this is typical for lesion of the inferior parietal lobule, but also for prefrontal lesion. Well, this is a case of ideomotor apraxia, which is a difficulty in making a given action under visual instruction or imitation, which stand in contrast to the fact that this action can be performed spontaneously by the patient. In this case, the patient was requested to make a military salute. Okay, he was doing like that. But he eventually, occasionally, over the street can meet a friend and do that. Okay? Just to tell that these are higher order form of motor behavior, and all these things do not happen after motor cortex. Fortunately. The first, the crucial evidence for cortico-spinal modularity is the metabolic cost of myelinated versus unmyelinated axons for generating one action potential. Now, this is a delicate issue. The metabolic cost for generating an action potential across evolution can be calculated as the energetic cost of discharging the capacitance of axons. Okay? Given the capacitance is the, in terms of electrophysiology, so in terms of nerve physiology, is a measure of the axon's ability to store electrical charges. and it plays a crucial role in the propagation of action potentials. There, the energy required to discharge a membrane is proportional to capacitance. And the unmyelinated axons have small volume, but very high capacitance, because they are bounded only by a single thickness of lipid bilayer. And they represent, as said before, a compact solution to the problem of maintaining connectivity while contributing minimally to space occupants, but they have very high metabolic rate, and therefore they are metabolically very expensive. While unmyelinated ones are exactly the contrary. And they represent a case which inform us that probably evolution have developed myelinated axons, which are a selected class of axons, just to compensate for the increase of brain size, and therefore to keep conduction distance delays in a reasonable range. So what you can see here, if you move from tissue to mouse, rat, cat, macaque, dog, and macaque, both the estimated metabolic cost and the measure one, which is performed through the oxyglucose studies, show that the metabolic cost decrease with brain weight. Okay, which is something which is coherent with what we have seen before when we have studied the how the proportion of myelinated fibers varies with brain size and weight. Okay, and this is so in this graph because of the prevalence of the relative prevalence in the macaque versus the tissue and the other animals of myelinated fibers. Now, these are the proof for cortico-spinal modularity. Now, I have the time. Is how much is for how much time did I talk?
If we can finish around 2, that would be good. Around 20 minutes.
Okay. Okay. Now I want to show you some example on the multiplicity and heterogeneity of processing stream and motor outflow system. And I want to, I would like, given the time constraint, to jump directly to one very interesting system in honor of Giuseppe Lupino, who has been done some work on this in some of this area, which is the ventral premotor spinal module that we consider as a model which binds feature for defensive behavior. Okay, and the ventral premotor spinal module is made as a crucial node, the ventral intraparietal area in the parietal cortex, and the ventral premotor area F4 in the frontal lobe. These areas are strongly and reciprocally connected. The ventral intraparietal areas receive a number of projections from the vestibular parietal areas, like parietal insular vestibular cortex, a parietal vestibular parietal sylvian cortex, and from some motion sensitive areas of the superior temporal sulcus, such as MT and MST. And these inputs probably confer the vestibular and visual motion sensitivity to this area. While a strong input coming from S2, probably, and also from inferior parietal area FP, confer somatosensory sensitivity to VIP. If we now move to F4, in addition to the projection from VIP, there is again a strong input from S2, okay, from area PF again, and from area 5 here, which confer somatosensory sensitivity. F4 project heavily to motor cortex, but the projection, the reciprocal projection, is not so intense. And the cortico-spinal projection of this model, beyond that, originating from motor cortex, which conduct at about 15 meters per second, is made by small axons, which convey information about 6 meters per second, less than half. So it is almost given for granted that they must play different roles. Okay. Now, which are the functional properties? Why this model is considered a model that binds different properties or feature for defensive behavior? Let's consider the areas which are involved, which I just named. Area MT, in area MT, which is area B5, neurons are sensitive to the direction of approaching visual stimuli. Okay? These visual stimuli can be directed, especially if they're directed toward the face, which is a very sensitive part. Imagine that if a stone or a ball come toward you. And they are also sensitive to the angle of gaze. In other terms, the activity to an approaching stimulus varies depending on where the animal is looking. In area VIP, the same author, Colby, Duhamel, and Goldberg, found a class of bimodal visual and somatosensory receptive field, which are especially common. In other terms, if the neuron respond to a visual stimulus, which is located near this part of the body, near the face, this neuron also will respond to the stimulus moving in that part of the body space, but could be on the end, could be on the body. And a similar property has been found in Giuseppe's lab by Fogassi, but also by Graziano, Princeton. Bimodal match directionally sensitive visual and somatosensory receptive field, but with the difference that in area F4 they are not sensitive to the angle of gaze. So these cells with specially aligned visual and somatosensory receptive field in parietal and ventral in VIP and area F4 are a potential near substrate for impact prediction and defense. And they do not simply reflect stimulus configuration, but they probably reflect action value. Action value intended in the reinforcement learning framework, if you want. Because we are talking about a defensive action, which have very high value, and which is important for shaping also the nature of our extrapersonal space. But what about the output? How do we intercept moving target? This is a brilliant experiment performed by Ugo Merchant, Alexander Battaglia-Meyer, and Apostolos Georgopoulos many years ago, where they train a monkey to move an isometric joystick as to displace a visual cursor from one position at the center to a position at 180 degrees, okay? The monkey was supposed to do that while a visual cursor was moved from 12 o'clock towards 6 o'clock, okay, at variable speed from trial to trial. And the monkey had to move his cursor to intercept the moving one within an angle no less than 18 degrees. Otherwise, the trial would have been aborted. Okay? In a different experiment, this is a real motion. They also tested the apparent motion. Now, in this case, there was not a stimulus moving continuously, but was flashed along the circle at different locations, giving an apparent motion condition. Okay? And the monkey had to perform the same task. In addition, and this was also moved at different speed. In addition, they performed a no-go task where the monkey was looking at the screen in both conditions, circular, moving, and apparent motion without doing anything. And in a center-out task, the monkey was supposed to make pulse force in a different movement direction, okay, to see what neuroactivity was. And you see that what they found is that in the no-go, in the center-out task, there was no virtual activity in motor cortex and in posterior parietal cortex in area 7A or PG, okay? And the analysis of the time-varying activity, okay, this is the moment in which the stimulus start moving, okay, in the real motion condition and the apparent motion condition. And this is the activity in parietal cortex. So parietal cortex fire earlier and motor cortex fire later. So the first activity track is a visual activity which track the stimulus motion, and then the output force of the monkey generate the motion of for the interception. But you can see that parietal activity also participate to the motion overlapping time with the output of motor. Okay, so the conclusion is that the population of motor and parietal cortical cell responded during the interception, okay, but not during the go and no-go task. And therefore, this group of cells can be considered specific for the interception of apparent moving stimuli. The time-varying activity, as I said, encode both stimulus motion and motor out in both the interception and in the apparent motion condition, in the real and apparent motion condition. While the activity which is related to the stimulus angle, especially in the apparent motion condition, predict the time to impact, which in the invertebrate is the so-called tau signal, which is probably only purely based on the expansion on the rate of an approaching stimulus on the retina, which when we pass a given threshold, start the escape movement of the locust. Okay, but this is a different kind of animal. And in addition, there is also, we'll see later, a gravitational component. So the idea is that all this information can be used in a predictive fashion to trigger the interception. Now concerning this issue, intercepting a target in real life implies a strong gravitational component, since gravity accelerates object, and therefore, if you are going to intercept a falling ball, your muscle and your brain make an anticipatory movement which predict the point of impact. While if you do this experiment on Earth, if you do this experiment on space, like this author did, Francesco Laquaniti and his people, on the shuttle space, when the astronauts that were accustomed to the 1G environment on Earth, when they arrived in space and did the experiment, of course, they were constantly anticipating the time to contact while the ball was slowly falling without an absence of gravity. This implied that whatever in real life stimulus we try to object we try to catch, avoid, we must take our brain contain an internal model of gravity that has to be set in motion. Otherwise, the task will fail. And the cortical network, which is engaged approaching visual targets which are accelerated by gravity, this is an fMRI study, include the posterior insula, the retroinsular region of the intraparietal cortex of the parietal operculum, and the region of the cingulate supplementary motor cortex. And the cortical areas processing a visual target, which are accelerated by gravity, are also activated by vestibular stimuli. And this is the vestibular connectome on the medial and lateral side of the brain. And you can see that the area, there are a number of areas which are somehow parallel or homologue, if you want, of the areas which have been shown to monkey as related to the defensive behavior and related to vestibular signal. And on the medial aspect of the hemisphere, you have retrosplenial cortex, cingulate cortex, and also the subiculum, because these areas are also important during spatial navigation, since vestibular information is very important during spatial navigation and locomotion. Now, let's come back to area V1 and F5. Spatial lines, somatosensory and visual receptive field as a neural subset for implant prediction and defense. If you microstimulate area VIP in the monkey with long-train microstimulation, and these are experiments done by Michael Tiziano, instead of eliciting specific simple movement, okay, like hand flexion and extension and so on, you observe a constellation of the movement which include eye closure, facial grimaces, so is a facial change, head withdrawal, elevation of the shoulder, movement of the head toward the mouth or eyes, or the space beyond the head or shoulder. In other terms, they are complex actions which are interpreted as resembling defensive behavior after F4 microstimulation. And the lesion study, which means chemical suppression of VIP through injection of mucimol, decreased defensive responses, while activation of VIP strengthened and exaggerated by injecting, for instance, bicugulin, defensive reaction. While lesion of F4, which has been done by Rizzolatti, and I don't know whether you were also one of the authors, Giuseppe, of this paper, lesion of F4 result in a peripersonal neglect. So that the animals seem to ignore what's happening around you. Now, let me try to introduce a little diversion. This is an image from a model that we made on the representation of extrapersonal space as a function of action value, okay? And in this case, you can have an action value which will get you to a reward, which is catching an apple. In this case, the action value is to escape, okay? And in one case, for catching an apple, you involve the nice Lupino's and colleague lateral grasping system from inferior parietal to inferior premotor cortex, okay? And these are the units, the network units of a reinforcement model made to mimic this kind of behavior. Since when in our extrapersonal space, often we face positive reward, but sometimes we also experience negative. Now, when the neuron to neuron distance, okay, is said to be the inverse of the neuron to neuron connection strength, you can see that clear goal and threat subnetwork or submodule emerge. And the emergence of this submodule improve the behavior, which is catching the apple and avoiding the snake, okay? So even the reinforcement learning model reshape your extrapersonal space in such a way to optimize this function. In other terms, the extrapersonal space in Rory Bufacchi and Gian Domenico Iannetti conceptualization, which in my opinion is the most advanced available, is not a fixed bubble around you that you carry with you in mobile, but is a 4-by-graded field which expand or contract depending on the value of action that you perform within it. And this has been shown by several fMRI studies, single unit recording, EEG studies, has been shown by behavioral studies, and now has been modeled by using a reinforcement learning procedure, where my contribution has been basically to help the setting the anatomy and let's say combining the anatomy and physiology, but then for what the modeling is supposed is all work by Rory Bufacchi. Now, lesion in humans. This peripersonal field, neural activity in VIP and F4 been tentatively linked to the perception of ownership of a location of body part and even of the whole body. And most of this work has been done here, as you know. So we interpret this data coming from this model in this sense as well. A reduced feeling of ownership of a body part and defensive response has been observed after lesion of VIP and the surrounding white matter. You don't see anything like that after lesion of motor cortex, okay? And damage to this area is also result in somatoparaphrenia, which is a condition or a misbelief that one's body part belongs to someone else. And this condition seems also to be associated with impaired defensive behavior. A different perspective, which has been put forward by Carl Friston, which is very interesting, come from the, let's say, it's an entropic perspective. which rely on defensive function. While our physiological function, like drinking, eating, urinating, imply very low entropy, their normal physiological function, a defensive function, is a function which aims at avoiding that our body is going to be damaged, dismembered, okay? And therefore imply a very high entropy. And therefore is a conceptualization of the defensive action are those which are characterized by very high entropy. Finally, for this model, least but not the last, coding of numerosity in area VIP and in ventral performance. Numerical judgment, which does not mean that monkey are able to count, but they can distinguish group of items or ensemble formed by a small group or large group. Therefore, you can imagine, I don't have to tell you how numerical judgment is crucial for our defense, for our defensive behavior. Let's imagine the situation. What would you do? Would you escape immediately, or would you stay, eventually to leave? Imagine a snake, okay? Now, should I stay or should I go? Now look at these two nice cortico-spinal projections. This projection here conduct action potential at very slow velocities. And therefore is the function of presetting the interneuron and eventually the motor neuron in a delay time, in a time interval, when in front of a threat, you have to decide whether you should stay, to think what is the best to do, and then decide, escape or attack, okay? I don't know if you remember that paper, Giuseppe, I've been unable to find, but there is a study by Jun Tanji that recording antidromic activity from F4 or recording, yes, from F4 neuron, found neurons which are tonically firing at low rate, which is what you need for postural adjustment, which are important for setting your defensive behavior and then decide what to do. But in any case, the conduction velocity is not the one that you would prefer to set an immediate reaction. While in this case, an interplay between this projection from F4 and the one descending from M1, which go at double the speed, is what you need. Once a decision has been taken. Now let's move to the third and final part of my presentation. It's too late. Okay, just a few minutes. Then what is the role of M1? The role of M1, taking a reinforcement learning perspective, is to serve as a model recombination when you learn novel tasks. Okay? In fact, a hierarchical reinforcement learning suggests that models offers a library of tools that can be dynamically recombined when you learn a novel task. Imagine that you have I was saying a monkey, a rat, a mouse, performing a limb motor task, which consists in moving when the LED of this object is illuminated, this object from a random location to the workspace to the center in order to get the reward. And then you have a second task where the object is fixed and there is an LED on top, and the water dispenser, the water spout is here. When the LED is illuminated, the animal has 2 seconds to lick to get the reward. Otherwise, the reward doesn't come in. So this is task 1, and this is task 2. And this is a composite task, which put together the 2 elements, okay? It has a, now the object is movable, and by using this joystick, the animal can move the object toward it with the water dispenser when the object is illuminated and lick. And if you compare the training rate in the composite task of animals who have not been exposed to task 1 and task 2, this is the training, the learning, the training when they have to learn from scratch. These are weeks, okay? While if you compare with those, with animals who have been pre-trained, you see that they reach the level almost immediately. You have an enormous advantage, okay? Which imply that if you simultaneously record calcium imaging from a number of cortical areas, which are M1, supplementary motor cortex, S1, posterior parietal, and retrospinal cortex, you derive the action value representation of this neuron, okay, where the action value is derived by taking into account the task state, okay, so LED on, LED off, the target at the center, target on the periphery, and the action, licking or moving the hand. So you compute the Q action, and these are the representation of the action value representation of neurons in the naive and the expert test in task 1, okay, and in task 2. And you see that the expert state, when the animal have learned, there have been a drastic redistribution of module in this area with an increase in the number of neurons which participate to the task. But when you move to the composite task, you see that from the early stage of the composite task, in the movement component, you have a distribution of Q value representation which resemble the expert late stage in the simple task. But when you have a transition from the early to the late stage, you again have a dramatic reorganization of modules, of neurons and modular interactions. So as learning progress in the composite task, a transition occurs from a dedicated to a more distributed representation, which led to a new policy, of course, composition and module recognition. Another set of studies suggests where this early banding of module may take place. In this case, M1 was activated in rodents. Oh shit! Was activated during foreign movement by injecting channel rhodopsin and labeling parvalbumin expressing GABAergic in the interneuron with a red fluorophore. And the results show that while M1 inactivation impaired the foreign movement during the early and middle stage of learning, he had no effect whatsoever on the late stage. And not only, but the M1 disengagement from the task improved the animal performance, showing that it's not a question, simply a question of level, but is a question of level of training, but is a question of expertise. But this can be seen not only in rodents, it can be seen in humans. by using both signals, okay? In this case, humans were performing a precision grip task. And in this precision task, you see that there is a decrease in M1 contralateral to the injection during the task. You have also a decrease in the frontal pole area and in dorsolateral prefrontal cortex. But at the same time, in the left premotor cortex, the activity remained decreased a little bit, but remained the same, while reach a peak and then decrease in presupplementary motor area and decrease dramatically in the intraparietal sulcus, which imply that the decrease in activity of motor cortex is accompanied by an increase or a differential kind of activity in other areas. In other terms, the functional connectivity between these areas changes. And this is shown in this slide, in which I don't want to enter into the detail, but you have an expert eye, and I can show you into later on. And even sleep-dependent improvement of early learning coincide with decrease in both activity of M1. So that M1 is bypassed during sleep by the recombinatory pattern which has been learned. And therefore his representation is somehow weak. And this is the pain. And this parallel the way the hippocampal sequences are used to consolidate memories in the cortex, while simultaneously weakening the sequence representation into the hippocampus. Conclusion. The M1 recombined signal in this hypothesis of M1 serving as a recombinatory hub must catch up, of course, with those which are addressed by the other modules, which are set in motion depending on the kind of function we are considering. Imagine defensive action that we are considering. To ensure that coherent command reach the spinal cord. But what I found by chance recently by looking at fMRI study, fMRI imaging of the spinal cord is extremely interesting. Since during motor task, imagine a precision grip task or a grasping task, the M1 spinal cord coupling, of course, you have activity in M1 and simultaneous activity in the C3, C4, C5, the relative spinal segment where the cortico-spinal projection is addressed, in this case monosynaptically. Over the course of training, this coupling decreases. Not only decrease the coupling between cortex and spinal cord, but also activity moves across different sectors in the spinal cord. Therefore, of course, within the same segments. If this is true, if this is also true, this modularity and plasticity, early, immediate plasticity for early learning are really at the basis, at the roots of action control. And this is all. Thank you.